Okay, so the enzymes that are used by the liver and by the muscles to convert glucose into glucose 6-phosphate are slightly different. So the muscles use a hexokinase, and now the liver uses a special hexokinase called glucokinase. So there's actually, like you can see right here, there are 4 isotypes of hexokinase, and most cells will use 1 through 3, but the liver and the beta cells of the pancreas use isotype 4. So isotype 4 is called glucokinase. Now the difference between these two enzymes is important, and if you don't know how to understand an enzyme activity curve like this, then I encourage you to go watch my videos just labeled biochemistry and the one's enzyme kinetics, and that will help you to understand this. But what we have here is the Km for hexokinase is very low, and the Km for glucokinase is very high. So what that means is that whenever glucose enters in the cell and binds with hexokinase, it doesn't stay bound for very long. It goes in, hits hexokinase, and becomes glucose 6-phosphate. Whereas with glucokinase, the molecule comes in, it interacts with glucokinase for a minute, and then finally becomes glucose 6-phosphate. And so it takes a little bit longer for glucokinase to convert glucose into glucose 6-phosphate, whereas it happens very rapidly in most other cells, especially in the muscle cells. It happens very slowly, or not slowly, but slower in the liver. And what this does is it allows the glucose level. So if we imagine that we have the blood here, this is going to be our blood, and then over here is our liver. And the glucose molecules, the concentration that's in the blood, should kind of match the concentration that's in the liver, because it's not getting converted into glucose 6-phosphate very quickly. Whereas if we have blood here, and then we'll say this is a muscles right here, the glucose in the blood here will be much higher than the glucose in the muscles, because it's being converted into glucose 6-phosphate so quickly. Now the purpose of this is because sometimes the blood glucose is going to be pretty low, and whenever it is low, the liver needs to be able to produce glucose and send it out into the blood. And now if it's producing glucose and then the glucokinase is turning it back into glucose 6-phosphate as soon as it's getting produced, then that would be a bad thing, and muscles wouldn't be able to get glucose. So by glucokinase having a higher Km, the Km, by the way, just as a reminder, is the Michaelis constant in enzyme kinetics. So by it having a higher Km, the liver and the pancreas can kind of monitor the concentration of blood sugar. And that happens because the amount of glucose in the liver and the pancreas will be very similar to the amount of glucose in the blood, because glucokinase is much slower than hexokinase. Now, glucokinase has one other important distinction from hexokinase. So hexokinase, imagine the glucose goes in, hexokinase converts it to glucose 6-phosphate. Then when glucose 6-phosphate builds up, it kind of feeds back and inhibits the hexokinase. But not so with glucokinase. Glucokinase is not inhibited by glucose 6-phosphate, because in the liver, glucose 6-phosphate can be used for several different things. It can be used for example, it can be used for glycogenesis. And so if you're wanting to produce more glycogen in the liver, you're making a whole bunch of glucose 6-phosphate for that, but you want to keep making it because you're going to convert some of that to glycogen, and some of that is going to go on down to fructose 6-phosphate. So in glucokinase, the fructose 6-phosphate concentration feedbacks and inhibits the glucokinase. So whenever it's going in this direction, once we get a buildup of fructose 6-phosphate and say, okay, we got too much now, let's stop what's going on over here. And this is also good because in gluconeogenesis, you end up producing a lot of fructose 6-phosphate and feeding back in that direction towards glucose. And so at that point, you can say, hey, we're going in the other direction, glucokinase, turn off, stop doing your thing. The last thing I want to say about glucokinase is that it's an inducible enzyme, so you can actually increase its activity, and one molecule that does that is insulin. So insulin signaling actually induces glucokinase to increase its activity, and that's good because once you have a carbohydrate-rich meal, you actually want to, your muscles aren't going to use all of that, and so in order to kind of buffer everything, the glucokinase will turn more of it into glucose 6-phosphate, and then it'll go over to glycogen. It'll be converted to glycogen. So the bad thing about that is whenever you have like a diabetic, the glucokinase in the liver, you're inducing whenever you're inducing glucokinase, you're increasing its expression, and there's less glucokinase being expressed in the liver, and so it hurts the liver's ability to buffer the blood glucose levels whenever you're diabetic. And also, so the enzyme that converts glucose 6-phosphate to fructose 6-phosphate is phosphoglucoisomerase, and it works at equilibrium. So there's almost an equal amount of G6P as there is F6P. And so to some extent, that mechanism can help because you're not going to have fructose 6-phosphate feeding back and inhibiting the glucokinase. So that can help whenever you do have low insulin levels, but still low insulin levels are detrimental to the whole mechanism of the liver. Not low insulin levels per se, but diabetic low insulin levels are. Especially when your blood sugar is high, you want your insulin levels to increase. Another cool feature about the liver is the GLUT2 transporter. So glucose goes into the cells into various cells by GLUT transporters primarily. There are more. They can use more than just GLUT transporters, but primarily GLUT transporters. But the liver expresses a special one called GLUT2, which is bidirectional, so it can go in or it can go out. Now GLUT2 is a high-capacity, low-affinity transporter. So what that means is sugar will go in through the GLUT transporter at a slower rate than it will go into the other tissues. So it's not competing. Everything about the liver is, I don't want to compete with the other cells for glucose, and so it goes in more slowly, and it can also come back out. And this is another mechanism that allows the blood glucose concentration to kind of equal the liver glucose concentration. So if it's going bidirectionally, if the blood has a higher concentration of glucose, it's going in the liver. But once the liver gets a higher concentration of glucose, it's going in the blood. So the glucose concentration in the blood and in the liver should always be pretty equal. Now let me just say really quick that I found this image on a Google image search, and from the textbook and from the lectures, these numbers are just a little bit off. So less than about 5.5 millimoles of blood glucose, that's when, according to the textbook, that's when glucagon signaling will start to increase. And so at less than 5.5, you would be over here on this side pushing out glucose, and at greater than 5.5, glucose should be going in. So 5.5 would be kind of the equilibrium between these two mechanisms.